In this presentation, we are going to focus solely on the digestion and absorption of the 3 energy-yielding macronutrients. Throughout the digestive system, the functions of digestion, secretion, and absorption occur. These processes are controlled by various hormones, such as gastrin, secretin, and cholecystokinin. Enzymes that are substrate-specific are secreted to digest the various macronutrients. Many times the enzymes end with the suffix ase, with the first part of the word indicating the substrate that is broken down. So the example on your slide there is sucrase, which breaks down sucrose. Once the nutrients are digested, they will be absorbed through the mucosal lining of the enterocytes. There are 2 basic transport systems: passive transport and active transport. The primary differences between them are whether energy is needed and if a transport protein is needed. Absorption occurs in one of 3 ways. First, we have simple or passive diffusion, which doesn't require energy or a transport protein. Some fatty acids and vitamin E are transported via this pathway. Facilitated diffusion doesn't require energy, but does require a transport protein. Water-soluble vitamins and fructose need a carrier, but no energy, as the nutrients are not moving against the nutrient gradient. Active transport requires both energy and a transport protein. Glucose, galactose, calcium, iron, and the amino acids all need a transport protein as well as energy, as the nutrients are moving against their concentration gradient. With this brief review of digestion and the basics of absorption, let's shift our focus now to the digestion and absorption of the 3 energy-yielding macronutrients. Carbohydrate digestion begins in the mouth with salivary amylase hydrolyzing a small amount of the carbohydrate. In the stomach, the activity of salivary amylase is deactivated due to the acidic nature of the stomach. The majority of carbohydrate digestion occurs in the duodenum, with pancreatic amylase breaking down the large starch molecules into disaccharides. The enzymes maltase, sucrase, lactase, and isomaltase are secreted in the brush border to break down maltose, sucrose, lactose, and isomaltose, respectively. Fibers are not digested by humans because we do not have the substrate-specific enzyme to break down the fibers. Fibers then pass through the small intestine into the large intestine, where the microbiota try to break them down. Some of the fibers will swell, some will ferment to form short-chain fatty acids and gas. The resulting monosaccharides of glucose, galactose, and fructose pass through the enterocytes and into the bloodstream via the blood capillaries of the villi, where they are carried by the portal vein to the liver. Glucose and galactose are absorbed by active transport, and fructose by facilitated diffusion. Since fructose is absorbed by facilitated diffusion, this slows its entry and produces a smaller rise in blood glucose. This can have implications for those with diabetes. Protein digestion begins in the stomach with the action of the enzyme pepsinogen, which is activated by the acidic nature of the stomach into pepsin. Most protein digestion occurs in the duodenum with pancreatic enzymes. Contact between the chyme and the intestinal mucosa allows for enterokinase to transform the inactive pancreatic trypsinogen into trypsin. Trypsin then activates carboxypeptidase and other inactive protease chymotrypsinogen into chymotrypsin. Proteolytic peptidases located on the brush border also act on the polypeptides, breaking them into amino acids, dipeptides, and tripeptides. The final phase of protein digestion takes place in the brush border, where all of the peptides are broken down into single amino acids. Amino acids then are absorbed via active transport, meaning that they need energy and protein carriers to move them from the brush border into the blood capillaries. Several different transport molecules are required due to the differences in the radical group of the amino acids. Some amino acids share carriers, so they will need to wait their turn before they are transported. Once inside the intestinal cells, amino acids may be used for energy or to synthesize other needed components. Those remaining travel to the liver. The majority of lipid digestion occurs in the duodenum, with a minimal amount of digestion occurring in the mouth due to the action of lingual lipase. This activity is more prevalent in infants due to the higher percentage of fat consumption. Very little fat digestion occurs in the stomach except for the role of gastric lipase in children. In the small intestine, the presence of fat in the chyme triggers the release of cholecystokinin, secretin, and GIP, which inhibit gastric secretions and motility and signals the gallbladder to release bile. The combination of the peristaltic action of the small intestine and the emulsification action of the bile reduces the fat globules into tiny droplets, making them more accessible to digestion by pancreatic lipase. The goal of fat digestion is to disassemble the triglycerides into monoglycerides, fatty acids, and glycerol, which are all the end products of lipid digestion. Water-soluble fats, such as short-chain and medium-chain fatty acids and glycerol, can be absorbed directly into the bloodstream via the hepatic portal vein. The fat-soluble fats, including long-chain fatty acids, cholesterol, the fat-soluble vitamins, and monoglycerides, need a little help. When the concentration of bile acids reach a certain level, they form micelles with other lipid components. Micelles are emulsified fat droplets formed by bile surrounding monoglycerides and fatty acids. This allows solubility of the hydrophobic fat globules in the hydrophilic digestive fluids and transportation to the intestinal cells. Upon arrival, the lipid contents of the micelles diffuse into the intestinal cells. Once inside, the monoglycerides and long-chain fatty acids are reassembled into new triglycerides. Within the intestinal cells, the newly assembled triglycerides, cholesterol, and phospholipids are packaged with protein into chylomicrons for transportation. The intestinal cells then release the chylomicrons into the lymphatic system, where they travel until they reach the thoracic duct near the heart. Chylomicrons, which are the largest and least dense lipoproteins, transport diet-derived lipids, mostly in the form of triglycerides, from the intestine via the lymphatic system. As the chylomicrons travel, they lose some of the triglycerides, thus getting smaller in size. VLDLs, or very low-density lipoproteins, originate in the liver from the remnants of chylomicrons that have been returned to the liver and from other sources of carbohydrate, protein, and alcohol. VLDL transports triglycerides for use, and as the VLDLs lose these triglycerides, it becomes more dense with a higher proportion of cholesterol. As the VLDL lose the triglycerides, it becomes a cholesterol-rich LDL, or low-density lipoprotein. The LDL makes its components available to the cell. The LDL is mostly cholesterol. HDL, or the high-density lipoprotein, originate in the liver to carry cholesterol from the cells back to the liver for recycling for disposal. HDL is mostly protein, This pathway is sometimes referred to as the scavenger pathway. So what happens to the bile? After bile has entered the small intestine and emulsified the fat, the majority of the bile salts are reabsorbed in the lumen and returned to the liver via the hepatic portal vein to be recycled back into bile that is then sent out to the small intestine again. This process is known as the enterohepatic circulation. A small percentage of the bile is excreted in the feces. Vitamin B12 also follows the enterohepatic circulation route. It is continuously secreted into the bile and delivered to the intestine where it is reabsorbed. Due to this recycling, healthy people rarely develop a deficiency. Most vitamin B12 deficiencies reflect inadequate absorption due to the lack of the intrinsic factor or a pH in the stomach that is not acidic enough. Both of these are a side effect of atrophic gastritis. This graphic is a simplified view of digestion and absorption as it takes place in the small intestine. To be absorbed, nutrients must be broken down and pass from the lumen into the mucosal cell lining of the GI tract. From there, the nutrient then either enters the bloodstream or the lymphatic system. Water-soluble nutrients and small products of fat digestion are released into the bloodstream and go to the liver. Larger fats and the fat-soluble vitamins assemble into chylomicrons. They then are released into the lymphatic system via the lacteals and enter the bloodstream nearer the heart, thus bypassing the liver first. Figure 1-9 on page 15 of your textbook provides you with a visual of the primary sites for secretion and absorption within the GI tract. As you can see, this graphic not only includes the macronutrients, but also alcohol, water, and the micronutrients as well.